So there are 6 steps to neural communication. The first step is resting potential. And so in resting potential, the sodium and potassium are inside the cell body. And so potassium is going to be inside and it's negatively charged, and sodium is on the outside and it's positively charged. The cell body is basically giving life to the cell, and the nucleus of the neuron is within the cell body. Resting potential occurs at negative 70 millivolts, and the sodium-potassium pump creates a resting potential and basically acts like a gate, which we'll eventually see later on in later steps. And then the dendrites, right here, they act as sites that receive signals from other neurons, and they're basically getting messages from other cells. So step 2 of the process is depolarization, and it's basically when you see the sodium rushing right into the cell body as potassium is rushing out. And this happens through the sodium-potassium pump, and the sodium and dendrites go into the cell body. Step 3 is threshold, and that's when enough sodium ions rush into that axon hillock, which is right here in pink. The sodium is able to rush through the cell body through voltage-gated channels, and the potassium leaves the cell body. Step 4 is action potential, and it's basically sending the message away so that the positive sodium ions go in quickly to create the desired domino effect that comes with this. The voltage-gated ion channels that are on the axon, right there, open when sodium rushes in. Whole process gets sped up by myelin sheath, which basically allows electrical impulses to move quickly and efficiently. And here's the myelin sheath right there. The nodes of Ranvier are ports not covered on the axon and are covered by the myelin sheath. So the nodes of Ranvier are right here, in here, in here. This process is called saltatory conduction, which is basically when the signal jumps from one node to another. So between steps 4 and 5, and between most of the steps throughout this process, is the refractory period, which is basically, it helps direct action potential down the axon because only channels downstream can open it and let in depolarizing ions. The neuron becomes polarized, and the sodium-potassium pump makes the sodium ions go outside the neuron. Step 5 is exocytosis, and that's when the neurotransmitters need to get released across the synaptic gap, which is right here. This occurs because of action potential. The electric message goes to terminal branches, and it shocks vesicles holding the neurotransmitters. The vesicle is moved to the membrane and attached to the cell membrane of terminal branches. It's right around here. And then some neurotransmitters that go into the synaptic gap attach to the ligand-gated channel in the dendrites and cell. So reuptake is the last stage of the process, and it's basically the neurotransmitters getting recycled by reuptake ports, which is right here, here. And reuptake is necessary because it's recycling neurotransmitters, which are really important, and it regulates the level of neurotransmitters present in the synapse.